DNA is the strand of genes located inside the nucleus. The genes inside the DNA are made up of adenine and thymine, and also pairs of guanine and cytosine. For protein synthesis to happen inside a cell, there has to be a step called transcription. Transcription happens inside the nucleus, and it starts at first when an RNA polymerase attaches to a DNA strand. As soon as it attaches, a long chain of mRNA starts to form along the unzipped DNA strand and code. Since the DNA inside the cell is the original and principal instructor for the actions taken inside the cell, it is important to have copies of this to not lose the original one. For this reason is that it exists the RNA and mRNA. The purpose of the mRNA is to copy the DNA strand. So the mRNA starts to copy itself by attaching to the unzipped DNA. So the cytosine component in the DNA will connect to a guanine component in the mRNA. A guanine component in the DNA will connect to a cytosine in the mRNA. An adenine in the DNA would connect to an uracil in the mRNA, and a thymine in the DNA would connect to the adenine in the mRNA. In mRNA, thymine is chained for uracil. So as soon as this process of transcription is complete, the mRNA strand is released from the DNA, and it goes through the nuclear pore into the cytoplasm of the cell. The third step of protein synthesis is translation. Translation happens outside of the nucleus, meaning in the cytoplasm. It happens when an mRNA strand gets out of the nucleus, and the ribosome attaches to it. Each amino acid is brought into the ribosome in a transfer RNA or tRNA. The type of amino acid in this tRNA is based on the anticodon of it. Base pairing occurs between the codon of the mRNA and the anticodon of the tRNA inside the ribosome. Codons are made by sequences of 3 nucleobases in the mRNA, and anticodons are made by sequences of 3 nucleobases in the tRNA. In the case of tRNA, instead of using uracil, thymine is used. As tRNAs connect and bond inside the ribosome, chemical bonds occur connecting the amino acids to create protein. Finally, the protein synthesis is completed. The amino acids connect to create polypeptide chains which produce proteins.